This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs.
My first speaker is Dr. Eliza Bandera, and she's an MD, PhD, and with a strong epidemiology with a strong interest in community health. She actually has served as a member of the American Cancer Society in 2006 on the Committee on Nutrition and Physical Activities Guidance for Cancer Prevention, in 2007 in the World Cancer Research Fund International and American Institute for Cancer of the World Cancer Research Fund, and setting a lot of the policies and helping, serving on numerous expert panels. Currently, she's a professor in the, I would just have to kind of read it out from here, is the Cancer Institute of New Jersey, and also associated with the Robert Wood Medical School, and right now is potentially changing its name to merging with potentially Rutgers University. So I think her affiliation might change very quickly. So with this very brief introduction, it's my pleasure to welcome her to give a talk on mycoestrogens in girls' growth and development. Eliza.
So I would like to, first of all, thank the organizers for the very kind presentation, very kind invitation to present my work. And of course, I would like to, first of all, acknowledge my collaborators in this work in New Jersey, and particularly Dr. Brian Buckley, who developed the assays, the methods to measure mycoestrogens, and conducted all the assays, and also Dr. Ian Marshall, who's the chief of pediatric endocrinology, among others. So I'm interested in the period of puberty. As we have heard this morning, is a well-known critical period of susceptibility, and we know that an early age at the onset of menarche is a well-established risk factor for breast cancer. And we also know that there's a major public concern right now with the falling age of puberty in girls, not only in the US, but also it's a global issue. And also, we saw this morning mentioned the Institute of Medicine report, Breast Cancer and the Environmental Life Course Approach. And I wanted to point out that the mycotoxin that I'm going to be talking about, zearalenone, was highlighted as a high priority topic. So what are mycotoxins? Mycotoxins are labeled as the most important contaminant in the food chain, and it's a secondary metabolite, a chemical of a fungus that produces toxic result in another organism. And what's important to note is that the fact that you don't see the fungus in the food doesn't mean that the mycotoxin is not there. When I presented this, sometimes people tell me, who will eat moldy bread? You will see the mold and you won't eat it. But the mycotoxin may be present and you don't actually see it. So keep that in mind. So fungal contamination can occur in many different stages during food production. It could be in the field, during harvesting, during storage, not only in the farm, but also even in your pantry. It's like any other fungal contamination or infection. It occurs most likely in conditions of high temperature and high humidity. And the spores may lay dormant for months or 2 years and waiting for the positive condition for and then they germinate. Important also to note that they're heat stable, these mycotoxins, so they're not destroyed by canning or other processes. So they stay there. This is just a diagram, again emphasizing the different stages during which the mycotoxin can be produced. But mostly what I wanted to show you here, you can see the humans there. So humans can be exposed to these mycotoxins through ingestion of contaminated crops, but also through ingestions of animal products from animals that consume the contaminated crops. So keep that in mind too. This is a list of some mycotoxins. So the ones that I'm studying are not the only ones. Some of them are very famous, like aflatoxins, which is a very common cause, a very well-known cause of liver cancer, for example. Just to put the whole thing into context. So I'm interested in seralainone, this mycotoxin that I'm talking about. And I want to point out that most of the evidence that we know is from experimental studies, so cell lines, experiments in rats, and there's some studies in pigs looking at the metabolism and that type of thing, but the evidence in humans is almost null. So seralainone is a mycotoxin produced for fungal contamination of grains, as I said, and the species is the Fusarium species. And they have been shown, again, in experimental animals to produce acute and chronic health effects. It has been labeled for this reason as a mycoestrogen, phytoestrogen, and growth promotant. And at the experimental levels in cell lines, it has been shown to be able to bind to the estrogen receptor alpha and beta, and to have a full agonist capacity in ER alpha and a misagonist antagonist in ER beta. So we know from the experimental data that they are capable of binding to the estrogen receptor biological effects. And we also know that affinity to the estrogen receptor is actually higher than other well-known endocrine disruptors, such as BPA and DDT, but lower affinity than other endogenous estrogens. Seralainone is a synthetic derivative of the mycotoxin seralainone that I've been talking about. And this is an agent very interesting that is federally approved agent in the US and commonly used in meat production in animals. And you can see here the cows with the pellets in the ear for the administration of seralainone. But it has been banned in other countries, including the European Union and countries and Canada. The estrogenicity of seralainone is very, very high and is comparable to the natural estrogen, endogenous estrogens, but also comparable to DES. And is much more potent than genistein and BPA. And it has been shown, again, in experimental studies to induce ER beta expression and stimulate growth in human breast cancer cells. This is just an ad for Ralgro, which is the brand name for Seranol. And you can see that, I don't know if you can see it from a distance, but basically the advertising says that increases rate of weight and gain in cows. So you will expect that if it has these effects in animals, it should have some effect in humans. Just to show you a little bit of the metabolism that what we know, again, this is in animals, we don't know in humans. So you have seralainone, which is the main mycotoxin that I was talking about, and it gets metabolized to alpha seralainol and beta seralainol, and these get metabolized in terms to alpha seralainol and beta seralainol, and then both to seralainone. And all these compounds, the evidence seems to indicate that they can metabolize to each other, but it's not totally known the mechanism. So there's a question mark there because we totally don't know. Again, this is to show you there are several studies across the world that have tried to measure levels of this seralainone in foods. And this, I took this a selection of the studies that were conducted in the US and Canada from this report, the WHO report from 2000. And you can see that it's very variable, but it's there in many of the samples. And these studies are not perfect because some of them, basically they don't have random samples of the foods. The methods are not totally known. Many of them have very small sample size, like 1 sample or 18 samples. But the important thing, I'm pointing this out so that it shows that it's there in the food supply. So what do we know about puberty in this compound? So essentially, this period, as we all know here, is very sensitive to estrogen stimulation because of delicate balance at that time. but we know very little about puberty. So the current evidence we have right now is the anecdotal reports in Puerto Rico and Italy that showed that at that point, that was in the late 70s, they found that there were epidemic of precocious puberty, and they attributed to the use of anabolic hormones in foods. However, the levels were not assessed. And then there are two small studies that have been published where they measure levels in precocious puberty girls, and some of them had levels. And so I'm going to talk a little bit about one of them that is normally referenced as the why sertralinone causes puberty. But I'm going to show you a little bit what the study is about. So essentially, this study is an Italian study. And again, I want to emphasize that both sertralinone and ceranol are regulated in Italy. So what they did is that they took two groups of girls, one in Veraggio and one in Pisa, from precocious puberty, that have precocious puberty, group A and group B, and then group C and group D are the control groups. So they measure calendar year, and they measure height and different measures and hormone levels. You can see, if you look at the 4 columns, that the group A, which is a group of precocious puberty in Viareggio, they were slightly younger from the beginning. Then they measured microestrogen levels, and they found that only in group A, 6 of the girls have detectable levels, and no levels in the other groups. Then they went on to classify the girls with microestrogen levels. They classify them microestrogen positive, and they compare CPP, so central precocious puberty girls with microestrogen positive, and CPP girls with microestrogen negative urines. Bloods. They actually measure this in blood. And you can see that at baseline, this slide is at baseline, the girls in group E were slightly shorter, which is similar to what we found in the Jersey Girl study, and I will present my data. And these were the levels of sertralinone in yellow that they found. So again, I want to point out that these are an adjusted analysis. And also I want to point out that this may not be the ideal group to evaluate the effects, because these are girls already with precocious puberty, and they were under treatment already with tritopterin. So it's not maybe the best group to study the effects. And then they looked at follow-up at 3 months of therapy with the treatment and 12 months of therapy. And then they found that the group that were microestrogen positive actually were taller and tended to be heavier. So they concluded from this that sertralinone is a cause of precocious puberty. And again, these are an adjusted analysis. So it's certainly very intriguing results that needs to be evaluated further, but I wouldn't say based on this that this is a cause. So what we did, I'm going to present now what we did in New Jersey in the Jersey Girl study. You can find this in PubMed. It's published in the Science of the Total Environment. So the Jersey Girl study is an ongoing cohort study of peripubertal girls. Recruitment sources that we've been using is pediatric practices, media, community recruitment efforts. And I have to say that the study started like a small pilot, and it received so much attention in New Jersey from the press. Everybody is fascinated. Part of it, I think, is the catchy name of the Jersey Girl study, so it received lots of attention. And it made me expand the study and go from a target area that I had in the Princeton area to expand it to all over New Jersey, given that interest. So the girls in the study are healthy girls, aged 9 to 10 years. They have to be New Jersey residents and living with the biological mother with no cognitive impairments, and both mother and daughter need to speak English. And for this analysis that I'm going to be presenting, we use cross-sectional data from the first 163 girls participating in the study. So data collection includes an eligibility questionnaire that we do, an appointment, during which we obtain detailed body measurements. We collect a morning urine sample, a saliva sample, and we assess Tanner staging by physician, but also mom, independently. We have a main questionnaire that includes questions, environmental exposures and early exposures and all those kinds of things, 24-hour recalls, and then we do an annual follow-up survey. So the dietary assessment are three 24-hour recalls conducted at least one during the weekend and one during a weekday, so to be able to capture the differences in intake. And initially, I didn't coordinate this around the time. So the diet in assessment with the urine assessment, I didn't coordinate. So half through the study, I started to do it. So that means that I have a subset of girls for which I have diet the day before urine collection. So that way I can look at 4 sources of these things. So I will present that data. And then for the rest of the girls, I have 3 dietary assessments at any time before the urine collection. So the average of the 3 dietary assessments is supposed to represent the usual intake of these girls. For the urines, the moms were provided a urine cup that we sent in a package, and then they were instructed to collect the first morning void of the girl, and then they will bring it to the appointment. Immediately after the appointment, we took it to the lab, where it was aliquoted and stored at -70 degrees. And at the time of doing the analysis, one of the aliquots was transferred to the Environmental and Occupational Health Science Institute for analysis. For this, I also wanted to point out that we only measure free and conjugated ceranol metabolites for this particular project, and this was limited to limited funding. So keep in mind this, because probably if we measure total microestrogens, the prevalence of exposure and the levels are going to be higher than what I'm going to be presenting here. We corrected for urine dilution using specific gravity. Data analysis, I won't go into details. Basically, we use the standard methods and non-parametric measurements when we needed to do that, and we estimated prevalence ratios using Poisson regression. So the main questions that we had, and again, I want to emphasize, this was the first study ever done in girls. So we had no idea if we're going to find levels or not. So it was a very, very interesting and fun, actually, study to do. So can these microestrogens be detected? And if they are, where are they coming from? What are the food sources here? And do they have an impact? So we got the data, and we started to look at it, and yeah, it was very interesting that ceranol was present in 55% of the girls, and total microestrogens in 78% of the girls. And you had there the means and the ranges, etc. I put in purple so that you could compare the levels that we found with the levels in the Massard paper. Keep in mind that my levels are urinary levels, the Massard paper is in serum, but you can see that we found higher levels of ceranol and higher level of ceranol, which is not surprising because in Italy it's restricted. So they shouldn't have found levels in Italy to begin with because it's regulated. Then we looked at the levels by certain characteristics. And again, this is a small study, so it's not definite, and most of the things were not statistically significant. But it points to certain things that certainly need to be studied in further studies. Like with age, not much difference. Interesting that African American girls seem to have higher levels. There were certain areas in New Jersey that had higher levels than another. And also it seemed to be higher with a lower education and lower income. And there seems to be an inverse association with BMI. Then looking at the food sources, no matter how we looked at it, with type of analysis, beef and popcorn always came as the most important predictors. And we looked at it because we had very detailed dietary recalls. We were actually able to look at what brands, what type of popcorn, and it was all kinds of popcorn, not just the microwavable, because you will imagine the microwavable popcorn sits in the bag for a while, so there were your perfect conditions for this fungus to be produced. But no, it was also in the ones that are prepared, like in the bags. So anyway, popcorn. And looking at total microestrogen, the same thing, beef and popcorn, the major predictors. The next step was to look at health effects, and I had the interest especially in growth and the onset of breast development. So the most striking result was height. There was an inverse dose response you can see here with height, where the girls with the higher microestrogen levels in urine were of shorter stature. So actually it went in the opposite direction that I expected. So it seemed to be more acting as an anti-estrogenic compound. And again, I want to point out, too, that the ones that came up when I showed the levels, Seranol, the ones that are the compound that is administered to the cows, the levels were very, very low. So keep that in mind. So what I'm showing here is mostly from the grain, the contamination, that mycotoxin that contaminates grain. Then looking at the onset of breast development, again, you can see that there's kind of a trend here where the girls with the higher levels of mycoestrogens are less likely to have reached the onset of development. And I put the age also so that you can see that the age is very similar in the three groups, so that couldn't be the explanation. And then the next step we'll do a multivariate analysis when I'm able to control for everything. So even adjusting for age, BMI, the year of urine collection, total isoflavone intake, and I also repeated analysis controlling adding beef and adding the age of menarche of the mother. And they didn't make any additional difference. So anyway, again, this shows that the girls with mycoestrogen positive girls that had detectable levels in urine, were less likely to have reached the onset of puberty. So the limitation of this study is that this is a small sample size relatively, and particularly for the food sources, I only have a subset of girls that had the urine and the diet coordinated to look at these things. But I want to emphasize, again, this is the first study that looks at these things. And what I want to point out that it seems like it's an agent that is worth taking a look at. In conclusion, what we found is that mycoestrogens were indeed detected in urines of girls in New Jersey, particularly Seranol, and Seranol levels were negligible. I'm not saying that we don't have to worry about the exposure of the animal products being contaminated with Seranol. We didn't find it, but it's something that certainly needs more study. The main source seems to be beef and popcorn. And what we found that compared to mycoestrogen negative, girls with mycoestrogen positive urine tended to be of shorter stature and less likely to have reached the onset of puberty even after controlling for BMI. So our findings overall seem to indicate that Seranol may have anti-estrogenic effects, perhaps by competing with the endogenous estrogens. And this is similar to effects that have been reported for isoflavones. So while our results are not conclusive, they raise important questions that need to be tested in larger, more diverse population, and importantly, using a longitudinal design, because our study was cross-sectional. So it's really a snapshot of these girls in a time of their lives when they're actually growing. So as far as I know, it could be that it's only delaying the age at takeoff, but maybe the effect with final height is totally different. So it's something that really needs more study. So our future plans, we have this pending application, working with Larry Cushy and the team from the Cygnus study, and some of them are here. We are planning on looking at the other markers, so this will be a longitudinal, so you actually look at growth rate, looking at age at thylarchy, age at menarche, body size, body composition, almost final height at age 15, 17, and growth rate. So we can actually look at the process, not just at a certain time, but actually the age at takeoff and age at peak height velocity, so looking at growth rate. And also we will conduct additional analysis in the Jersey Girl study, and we want to compare levels in California and New Jersey. That may also lead us to some clues. Lots of people to thank in this study, especially all the recruitment and the interest in the community. But of course, I have to thank my daughter with the inspiration from the study. I started the study based on my interest, not only as a scientist, but also as a mom, because she was going through this puberty period and I wanted to know answers. What should I do for her? And now she's a beautiful teenager. So thank you.
Thank you. Okay, so this is a very exciting talk. I'm sure you will have tons of questions, but we're going to hold them.
Okay.
Thank you. Frank, okay, let me introduce my next speaker first. Okay, so this is Dr. Frank Burrow. He got his MD at Harvard Medical School and is currently the division chief for the adolescent medicines at Cincinnati Children's Hospital. And actually, he is among the founder for the puberty study at the Cincinnati site. First of all, it initially started out as the Breast Cancer Environmental Research Center, and then now is the program. He's the program PI, and he really has a lot of very distinguished career, but I know that Frank has an exceptional talent, and in his spare time, he's actually performing a lot of actings, and we got lots of his wonderful pictures on the web and all that. So I highly encourage you to Google him right now. So with your smartphones. So Frank is yours, and you have titles on already.
Okay. I apologize. Frank's not able to make it today, so I'm his brother, Michael, and I'll be presenting on behalf of the BCRP team. And I'd like to thank the meeting organizers and also all the people who work in the BCRC and the BCRP. The data that we're going to be discussing this morning could not have been generated without everybody working together. I'd also like to thank the institutes, NIHS and NCI, for their generous grant support to allow us to present these data to you. What I'm going to do is very briefly go through windows of susceptibility. We already discussed that this morning. We'll discuss the risk of breast cancer with different life events that occur in the childhood and adolescent period. And we'll define what puberty really is and introduce some data on pubertal milestones from the BCRP. This presentation is going to be integrating several of the other talks that you've already heard this morning and you'll be hearing this afternoon, because after all, it's all about puberty anyway. Okay, I borrowed this slide. This had been developed several years ago from Drs. Fenton and Dr. Russo. In the upper half, Drs. Fenton and Russo discussed the windows of susceptibility for breast cancer, and they discussed things that occur at birth, around the time of puberty, pregnancy and lactation, and menopause. And below the line, what I'm doing is showing you what are some of the pubertal events that are going on. And you're saying, wait a minute, below the line you've got birth. That's because the first puberty actually occurs in the third trimester. Fetuses, third trimester fetuses have a fully functioning hypothalamic pituitary ovarian axis. And it gets turned off in the first 6 to 12 months of postnatal life, that is, after they're born. We don't know exactly why it gets turned off, and it stays off then until the girl hits puberty. Below the line, the next thing is adrenarche. This is now the adrenal androgens that are being produced by the adrenal gland. And there's a change in the adrenal cortex in which there's not only anatomic changes, but there's biochemical changes, and they start now producing these adrenal androgens. After that, an undefined time after that, you have the reactivation of this hypothalamic pituitary ovarian axis. We don't know everything that leads to its onset, but we know that leptin, a hormone that's produced by the fat cell, is necessary but not sufficient to go into puberty. It appears that the signal to go into puberty might be mediated through another hormone called kisspeptin. And so with leptin and kisspeptin around, there's the reactivation of the HPO axis. Shortly after the young lady starts having breast development, there is the occurrence, there's a prepubertal growth spurt that actually precedes the onset of breast development, but the peak height velocity is then achieved after the girl has gone into puberty. And shortly after peak height velocity is attained, the girl has menarche. At the same time, almost 2/3 to 3/4 of a woman's bone mineral that's ever going to be deposited in her skeleton does so during this time of puberty. So you can see all these dramatic changes that are occurring in the human organism during the time of life that we call puberty. And the importance of these we're going to be going over in talking about some of the concepts of the development onset of health and disease. Again, we've already had several speakers talk about this metabolic imprinting and epigenetic changes that occurred during these critical periods of life. We heard about the Barker hypothesis. And again, this was based upon Dr. Barker noticing in epidemiologic studies that infants that were born small for gestational age, this is again independent of the relative timing, but rather these were born small for whatever the gestational age is, that they had an excess risk of several different adult outcomes, including hypertension, heart disease, and obesity. And there's been several different theoretical frameworks that have been offered for that, and part of it has to do with the in utero environment. What the fetus is seeing is felt to be, by the fetus, to be predictive of what's going to occur in postnatal life, that is, after the fetus is born. And what happens is, in this uterus in which it appears that there's not enough resources around, the body makes these changes, part of it through metabolic imprinting, part of it through epigenetic changes that lead to increased allocation for acquiring energy and storing energy, and actually relatively a large amount going for maintenance of brain growth. And this is done at the cost of repair mechanisms as well as overall somatic growth. Therefore, the babies are born small for gestational age, but typically their head circumference is maintained. So let's talk a little bit then about what are some of these life factors and breast cancer risk. The association of reproductive factors with breast cancer, I'll show in the next slide. But this is going over now studies that have been published in the last 20 years, looking at various life events that occur in this pubertal time period and the risk for breast cancer later on. So that there was a paper that showed that taller women had a higher rate of breast cancer, a higher risk of breast cancer, and those that had an earlier age of peak height velocity also had a higher risk for breast cancer. And we know that those who are tall have higher levels of hormone that's related to growth hormone. We know that there's a relationship between IGF-1 with breast cancer and with leptin, higher levels of leptin with breast cancer in adult women. And in adult women, we also know that there's an inverse relationship of another hormone produced by the fat cell called adiponectin with breast cancer. In adult studies, there's relationships with body mass index, that is how relatively heavy somebody is, and especially with adult weight gain. And last is that there have been 4 different studies that have shown longitudinally that women with a greater bone mineral density have a higher risk of breast cancer. And again, when is most bone mineral accrued? It is during puberty. The reproductive factors that are associated with breast cancer include age at menarche, and again, a decrease in 9% of premenopausal breast cancer and 4% for postmenopausal breast cancer for every higher age at menarche. There's been several studies that have looked at the number of cycles before you have first full-term birth. And lastly is the number of lifetime cycles, and contrasting those with less than 400 and greater than 500 cycles. Again, obese women have a lower rate of premenopausal breast cancer, but a higher rate of postmenopausal breast cancer. Obese women have fewer ovulatory cycles. So, looking at that now as a background and a framework for discussing puberty, what exactly is puberty? So puberty, I would maintain, is a reactivation of this hypothalamic pituitary gonadal axis and the development of secondary sexual characteristics. Those are the 2 traditional definitions for puberty. There's some recent data that suggests that perhaps these are 2 different biologic events. With puberty, you have an acceleration in your growth velocity, and you achieve the pubertal peak height velocity. There's profound changes in body composition in girls, and as a clinician, something that we have to deal with, the achievement of fertility. Let's define a little bit some of the concepts of puberty. So adrenarche, as we discussed, is this activation of the adrenal cortex and the production of adrenal androgens. Pubarche, pubic hair. Thelarche, breast tissue. Gonadarche, the appearance of secondary sexual characteristics such as breast development and the production of sex steroids. And menarche is the age of the first menstrual period. Take a look at the sequence of pubertal events in girls, and this is data that was generated from the NHLBI Growth and Health Study. 2,500 girls that were followed from the ages of 9 and 10 a couple of decades ago. And curve that you see is the pubertal growth spurt, and you can see that the pubertal growth spurt actually predates the onset of puberty or the appearance of breast tissue. About 1.5 years or 2 after that, the girl will achieve peak height velocity, and 6 to 12 months after peak height velocity is attained, the girl experiences menarche. The completion of puberty is defined by the attainment of breast stage 5 or pubic hair stage 5, whichever is later. But there's continued growth that occurs both after menarche as well as after the completion of puberty. This is looking then at some data from the Growth and Health Study with some historical studies. And these historical studies are from the 50s these were reported from the 50s into the 80s. And so, the correlation is looking at the onset of breast development with the age of the peak height velocity. In the Growth and Health Study, it was 0.27. In the historical studies, it was higher. The interval between the onset of breast development to peak height velocity is 1.7 years in the Growth and Health Study. Breast development to the age of menarche, and a lot of people will talk about this as the tempo of puberty. The correlation 0.37, the duration almost 2.5 years, and peak height velocity to menarche. In the Growth and Health Study, this was the highest correlation, and actually in a lot of the historical studies, this was also the highest correlation. That is, a high correlation between peak height velocity and age of menarche. But this correlation between the onset of puberty and the age of menarche has gone down. He's looking at studies that go all the way back to the 1930s and comparing to numbers 6 and 7, the Ritter reporting from Belgium and the Growth and Health Study. And you can see that this correlation has gone down, suggesting that the onset of pubertal maturation and the age of onset of breast development has become increasingly two different pubertal events. We've submitted this for publication, and this is examining data from the BCRP. And what we've done is we've compared and contrasted girls from our study to those from the pediatric research and office settings from Marcia Herman Giddens. The PRO study is the solid blue line all the way on the right-hand side. The three lines that are to the left of that are going from right to left. The dashed line is the girls who are below the 85th percentile for body mass index. In other words, the thinner girls. The dash and two dots represents I'm colorblind, so I cannot tell you what the colors are. My brother Frank can tell colors, I can't. The dash and the two dots represents the distribution of onset of breast development for the group overall. And then the line all the way to the left-hand side is looking at the girls who are greater than 85th percentile for body mass index. In other words, these are the overweight and obese girls. So our girls, the white non-Hispanic participants in the BCRP, are maturing at younger ages than that reported by Herman Giddens. And that it appears that a large contribution to this change is the increase in body mass index. When we did the analysis looking at the factors that are associated, in addition to race and the age of the participant, the other large factor that came out was body mass index. In fact, body mass index by itself accounted for 14% of all the variance. Now, people are going to say 14%, that's not much at all. 14% as a source of variance is huge and massive. Now, some of this could be accounted for directly by the impact of BMI. Some of this could be impacted by the effects of some of these exposures in our environments leading to changes within the fat cell. Dr. Birnbaum discussed the issue of obesogens. So it could be the impact of the obesogens on our body mass index. This is taken out of just the Cincinnati subset. The data are similar for the other two sites of the BCRP. But I had the opportunity to look at this. And we also just finished our year 2 of data collection for the BCRP after the 6 previous years of data collection for the BCRC. So almost all of our girls have had breast development. Actually, that little bar there represents one participant. Most have had pubic hair development, and about 70% of the girls in our study have had their first menstrual period as well. The girls who are premenarchal, that is, have not reported their first menstrual period, their mean age is actually younger overall than the girls who have had their first menstrual period. So it took us 2.5 years to recruit the cohort, so these are our younger girls in the cohort overall. So what I'd like to do is I'd like to close and then look at what we've been doing to human biology over the last couple or 3 decades. And below the line are the growth and health study girls. Again, 2,500 girls that were recruited from Richmond, California, from Cincinnati, Ohio, and from Washington, D.C., and contrasted them to the girls in the BCRP who were recruited from San Francisco, California. If any of you took the BART, you know that Richmond is just a little bit north of here. Cincinnati, Ohio again, and instead of Washington, D.C., now we're looking at New York City, specifically Harlem. And if you're taking a look at it, we can't give you the age of onset of girls for adrenarche in the NGHS girls, and these are actually written along, those dots represent actual ages in years. So adrenarche is clearly before gonadarche, that is the appearance of secondary sexual characteristics and sex steroids. What I'm doing here with gonadarche is, so gonadarche is coming a little bit more than a year after adrenarche, and about 6 to 12 months before the appearance of breast development. If you flip down to the lower one, you can see that similar to the PROSE study, that we're well ahead of where the age of thelarche was in the NGHS girls. We have an established age of peak height velocity for NGHS girls. We don't have it for the BCRP girls because they haven't hit adult height. It's very difficult to estimate peak height velocity without knowing where the adult height is. And I put there an estimate for menarche for the BCRP girls. It's advanced a little bit. But the point that I want to make is that we've dropped the age of B2 and only slightly decreased the age of menarche. We have expanded this window of susceptibility for puberty over the past 2 to 3 decades in our girls. That means that they are biologically at greater risk because of a longer duration to these other environmental compounds. I'd like to thank you so much for your attention this morning, and have a great day.
Thank you. This has been wonderful. Thank you for keeping the time. So that gives me a great pleasure to actually introduce my next speaker. And my next speaker is Dr. Esther John, and she is in the Cancer Prevention Institute of California, a senior research scientist and Cancer Prevention Institute. She's also a consulting professor at the Division of Epidemiology, Department of Health Research Policy, Stanford University School of Medicine, and a co-leader in the program of Cancer Epidemiology Population Studies at the Stanford Cancer Institute. I will give you all the time. Thank you, Esther.
Thank you very much, and thank you for inviting me. So my talk is going to be a little bit different. I'm not going to be presenting any results, but I will be introducing you to a new study in girls that we started last year. And I hope that we can come back, not just me, but the entire investigator team in future years and actually present results of what we're finding. We are going to be, I'll be presenting the Legacy Girls study. And I hope with this study that we can answer some questions about how young girls can reduce their risk of breast cancer. The Legacy study is a new cohort that we started last year in early 2011. We were funded for 5 years by the National Cancer Institute. And what we are doing is we are enrolling 900 girls. They are between the ages of 6 and 13. And half of the girls are the daughters of women who are enrolled in the Breast Cancer Family Registry, and I will talk about that in just a minute. And then for comparison, we have half of the girls who are for families without breast cancer. Recruitment is taking place at 5 sites in New York City. Mary Beth Terry is the principal investigator, and she's actually here together with her coordinator, Ann Johnson. We have a site in Ontario, Canada, in Philadelphia, Salt Lake City, and the San Francisco Bay Area. And these are the sites where the Breast Cancer Family Registry is located. What we are doing is we're going to be following the girls prospectively for 5 years, and we will collect data on biospecimens at 6 months intervals. So that's just for background, what we're planning to do. So the question, why do we want to study girls from families that have breast cancer? As has been long and well-established, having a family history of breast cancer is a risk factor for breast cancer. The risk is increased 2 to 4-fold depending on the number of affected relatives and relatives' ages when they were diagnosed. And the risk is much, much greater, 5 to 8-fold increased depending on whether a person is a carrier of mutations in the BRCA1 or BRCA2 genes. And therefore, girls who have a family history of breast cancer, who have relatives with breast cancer, are at increased risk. And this is what we're trying to focus on in the Legacy Girls study. Dr. Bandera has just nicely shown there is a number of early life exposures and events that we now know that increase the risk of developing breast cancer. So for us, we're hoping in the Legacy Girls study to answer the question, how can girls who have a family history, how can they reduce their risk of developing breast cancer once they reach adulthood? As I mentioned before, we are building this study upon the Breast Cancer Family Registry, which is a program we started in '95 in the US, Canada, and Australia. And since '95, we have enrolled over 13,000 multi-generational families into the family registry. And we oversampled young women with breast cancer, but we went all the way up to age 64. And because we have because of the oversampling, because we have many young women in the breast cancer family registry, we actually have many daughters that are not part of the family registry, but we are now enrolling them in the Legacy study. And the families that we have in the registry represent families across a broad spectrum of risk. We have families where there's only one person with breast cancer. We have families where there's multiple cases of breast cancer. And we have families where one or more members have mutations in BRCA1 or BRCA2. And so, as we are now looking at the daughters in these families, they have relatives with breast cancer. That can be the girl's mom, it can be the girl's aunts or grandmothers or other relatives that have been diagnosed with breast cancer. Why do we want to study early life exposures? Why is that important? I think we learned a lot about that this morning. And most of the studies that have looked at early life exposures have focused on adults, on adult women, and asked them questions about early life exposures. And based on the literature, what other people have found, what was presented this morning, we know that ionizing radiation, particularly at a young age, increases the risk of breast cancer. We just saw some data about early menarche, which is a well-established risk factor. We also know from migrant studies that in Asian and Latina immigrant women who migrate to the US at a young age, they have a higher risk of breast cancer. We think that's related probably to changes in lifestyle and reproductive factors as they start living here. A number of prenatal factors have been associated with breast cancer risk, things like birth weight, twin status, certain pregnancy conditions. Also, a number of childhood and adolescent factors have been associated with breast cancer risk, and these are things like the growth rate, body size, physical activity, dietary intake. So we're trying to put these two observations together. We want to study these early life factors in young girls, because if we study adult women and ask them about early life exposures, it's difficult to do because there's many years back to remember and the recall might not be very accurate. And if we are studying, let's say, young girls and we want to follow them until they might develop breast cancer, it can take many, many years because breast cancer still is fairly rare in women under age 40, let's say, but as the women get older, the risk increases. So our approach in the Legacy study is that we're going to study young girls, we're going to study early life exposures, but we're going to look at intermediate markers of breast cancer risk. And on the bottom of the slide here, you see I've listed the pubertal, the intermediate outcomes that we want to study. So we're interested in pubertal development, We're going to be assessing Tanner stage. We're going to be asking about age-up menarche. We're going to be looking at breast tissue characteristics using a novel method. We will be studying markers of epigenetic changes, and we will also study the psychosocial impact of increased breast cancer susceptibility. And these are outcomes that really reflect the interests and expertise of our investigator team. And on the last slide, I will show you who all the participants are. So we're going to be looking at these intermediate outcomes in green on the left side. I'm listing some of the early life exposures we're going to be studying in the girls. We're going to be asking about prenatal exposures and birth characteristics. We will study body size and growth, certain lifestyle factors like physical activity and diet. We will also be studying the social, family, and the built environment and selected biomarkers. And as you can see, I'm not listing environmental exposures on this slide, and I know this is something of great interest to this meeting and to this group of investigators. When we wrote the grant, we really had to focus our grant proposal. You know, you cannot propose to study everything. We are now have the opportunity, because we follow the girls, we contact them every 6 months, so we have opportunity to collect more data. And we, in fact, in our second follow-up study, we have now included some questions on environmental exposure. So we will be covering that as well. So what's particular to our legacy study? We are very much interested in finding out whether the effects of early life exposures operate differently in girls that are at higher risk of breast cancer because of their family history. We want to know, are the associations between these early life factors I showed you and the intermediate outcomes, do these associations differ? Are they modified by family history? And as we follow the girls over time, we would like to learn whether the rate of development or the rate of change in the biomarkers that we're studying, whether these are modified again by the family history. So to do so, we have, as I said, we have two groups. We have girls from the Breast Cancer Family Registry, the daughters, and then we will studying girls without breast cancer in their families, and they are at average risk, and that will be our comparison group. As far as we know, the Legacy Girls study is the only youth cohort worldwide, and this cohort will be enriched with girls at increased risk of breast cancer. They will come from a family that covers a wide spectrum of risk. And if we were to look in other cohorts, we might only find maybe 5% of girls who have a family history. So in order to study that, we really had to go to the Breast Cancer Family Registry, where we can find girls at increased risk. We will be looking at pubertal development, Tanner staging. We'll be looking at age-up menarche. And here I'm just going to show very briefly a few endpoints that we will be studying that are novel, related to pubertal and adolescent development. So they include breast tissue characteristics, the markers of epigenetic changes, and then the psychosocial impact of increased breast cancer risk. First, just a few words about breast tissue characteristics. As we know from studies in adult women, having dense breast tissue measured by mammography is a strong risk factor for breast cancer. Risk is increased 4 to 6-fold in women who have dense breasts, but we cannot do mammography in young girls to assess breast density because, as I said before, young girls are very sensitive to ionizing radiation. So we're going to be assessing breast tissue characteristics using a new method. It's called optical spectroscopy, OS, and this is a non-invasive method of assessing breast tissue composition. And what happens is a light is shined on the breast tissue, and it does not involve ionizing radiation. And the light absorbance then provides information on the breast architecture and measures certain components. This work will be led by Julia Knight and by Lothar Lilge, who is actually the person who developed OS. They are up in Ontario, where we have one of the legacy sites. So they will lead this work on breast tissue characteristics. Julia Knight has published work, and she has shown that OS can detect changes in the breast tissue that occur with age and changes between paras and nulliparous young women. So she did work in young women, and we're now going to use this method in our girls. The Ontario site is currently doing pilot work on this method, and then in years 4 and 5, all of the legacy sites will perform OS and will be doing this on girls who will be 10 years of age by that time. Another novel endpoint will be looking at genomic DNA methylation. Epigenetic changes may explain how individuals with identical genetic profiles have different health outcomes. We heard about that a little bit more this morning. This work will be led by Mary Beth Terry and Regina Santella from the New York site of the legacy study. Dr. Terry has done some pilot work in the legacy study, and they have shown that girls with a family history have lower overall levels of DNA methylation compared to girls without a family history. And lower levels of DNA methylation has been associated with in adult cancers and also with breast cancer. And then lastly, I will just say a few words about our aim that will address the psychosocial impact. Psychosocial well-being, including the performance of health and risk behaviors, has not been studied among young girls with a family history of breast cancer, so we have the opportunity to do so in the Legacy Girls study. And we have questionnaires to assess the psychosocial impact and hope to identify parent and daughter barriers and enhancement to adaptive behavioral and psychosocial responses. Currently, it is not known how young girls with a breast cancer history adapt to familial risk, and we don't know how familial risk impacts behaviors throughout the pubertal development period. So we think that the legacy cohort will be important in giving some answers to these questions. Now, this work will be led by Doctors Angela Bradbury, Mary Daly, and Linda Patrick Miller. They are at the Philadelphia Legacy site and will lead this work. So where are we right now? We are in the first year still of recruitment. We're about 80% done with recruiting the girls. We enrolled as many girls as we could from the Breast Cancer Family Registry, but also had to go to other sources because the Breast Cancer Family Registry women are, they're aging, so we have fewer and fewer with girls that are between the ages of 6 to 13. So we identified girls through other sources, other breast cancer clinics. And then we have the girls from the community, and we have identified them through friend referrals, through community outreach and community events. It has been a challenging enterprise to get these girls and have them wanting to participate for 5 years, but we've really found a lot of interest and have had really good follow-up. We're now in follow-up 2, so it's very reassuring that the families want to come back and continue. So what we do is we collect a questionnaire. We have questionnaires that we administer to all the moms or guardians. And we have questionnaires that we administer to the girls once they reach age 10. So for the younger girls, we don't collect any data. And we collect biospecimens. We collect blood or saliva samples and urine samples from all the girls of all the ages. And so we're building a very valuable repository of biospecimens to answer our questions, but also to have these resources available to answer future questions. We take body size measurements. We measure height and weight and hip and waist circumference. And we get information on Tanner staging by self-report, where the girls report and the moms report. And at 2 of our clinical sites, we also have nurses doing it. And then in the older girls, we'll get the OS measurements. And at this time, because we started recruitment last fall, so we have some girls that are now doing already the follow-up at 6 months, and we have some that just started at 12 months follow-up. And we're going to be seeing these girls every 6 months. So this is my last slide. This work involves a large investigator team and would have not been possible without the expertise, the broad expertise from a very diverse and multidisciplinary team. You see here the 5 sites participating in the legacy study. And we're also organized that each site is responsible for a particular core activity according to their expertise and interest at their site. And we received funding from the National Cancer Institute and greatly appreciate that, particularly now that it is so difficult to get research funding. And we feel very fortunate that this is a cohort study because at the moment it's difficult to get funding for new cohort studies. We were funded through 4 linked R1s, and I'm listing the grant numbers if somebody's interested. So this concludes my talk, and
Okay, so before I think we can open up for all questions. I would like to invite the last 2 speakers to come up here. We can have a panel discussion here. I think that may be able to answer both specific questions as well as collective questions that I think a lot of us must be waiting for a long time. And there are some sentences here. I think people are lining up for questions, but before you can actually answer questions, I have a special design for this panel discussions. What I like to do is to offer opportunities first to our community participants first, as well, and the students, as Frank was correct. So in that order, I would ask, the first question has to be a student. If you don't ask the question, you're holding us up. We have exactly 25 minutes. So one of you have to ask a question. There's no questions that are called, I was always told there's no questions that are called dumb questions. So please, then we'll take one question from the students to open up this format. Please.
Hi, I'm Elena Catan, and I'm here with the Breast Cancer Coalition from Great Neck. And I have a question for Dr. John. You said that, how come you guys don't use sonograms for the younger children to test them?
that we're not using what, sonograms?
Yeah, you said you're using...
ultrasound?
You can't use mammograms, so why don't you...
Yeah, we're not doing mammograms because when women or girls or young women are more sensitive to ionizing radiation, even so mammography involves very low levels. It would not be an appropriate thing to do with young girls. So we're trying to measure breast tissue characteristics in another way that does not involve ionizing radiation.
It's about ultrasound. Yes.
Oh, ultrasound. Oh, I'm sorry. We did not consider ultrasound for this project.
What is the difference between an OS and ultrasound in terms of the sensitivity? You phrased a very good question. I appreciate that, but that's what you're asking, right? So.
Yeah, how come don't you use the ultrasounds, sonograms?
Yes, as I said, we have not considered doing ultrasound on this group of girls. It's potentially something we could do as we do follow-up and the girls, get older. But at this time, it's not part of the protocol.
Okay, so is it to your satisfaction? And I saw a lot of hands raising. Are you an advocate?
I am an advocate.
Great. So why don't we take 2 questions and then we'll allow the scientists to ask some questions. So please.
So I'm here with the Great Neck Breast Cancer Coalition.
Yes.
And the thing I was wondering about is that Dr. Bandera was talking about the plastic urine cups that are used for collection. And I'm just wondering if there's any concern about contamination of the samples with the chemicals from the plastic?
We tried to use, we considered that when we were selecting the caps, so actually they were phthalates free, they were supposed to be, but I don't think that that should affect the levels of mycoestrogens. It shouldn't affect what I presented, whether the caps had phthalates or not.
But how do you know? I mean, it shouldn't affect, but have you tested whether or not? I guess I'm just wondering because we have students who have gone to the lab at Tufts University where they're studying BPA and breast cancer, and they sort of stumbled upon the fact that the plastic petri dishes were involved in they changed the results.
Again, when we were selecting the supplies, we actually took that into account. So I cannot tell you that we actually measure phthalates to respond to that, but they're supposed to be phthalate free.
Thanks.
Okay, I'm going to now give equal opportunities now.
So my first question is to Esther John. You mentioned that you'll be using the optical spectroscopy to measure breast density in the young girls. And so the methodology seems exciting. And my question is, has this been used in places other than the Ontario group, or is this a very new methodology being used?
It is a new methodology, and it was developed in Ontario and is being used right now in Ontario. I think there might be some other groups that are interested in it, but I'm not aware that anybody else is using that method right now. I'd have to find out from the Ontario team.
Okay, and my second question was about the Jersey girls study, I guess, Eliza Bandera. And my question was, one of the criteria that you used in including the young girls was that they needed to have the ability to speak English. And my concern was, would you be missing out on an important minority population if you do have that as a criteria?
Yeah, I agree with you. This was, again, I had limited funding and I didn't have the staff to translate all the material. So that's why for this particular study, we limited to English-speaking people. Again, this is an area that really needs further investigation, looking in minorities and that type of thing. So this is something that we're going to be doing in the future.
We're going to take a question from the front.
Yes, I am Paolo Toniolo from New York University School of Medicine, currently emerging from the water. It's a general question to the panel. I haven't heard any comment on the importance of the microbiome, which is now becoming probably the hottest area of research, and the fact that most of these girls or young women will have been exposed to sometimes many cycles of antibiotics during their life, certainly will have had an impact as we now start appreciating how that is relevant. And perhaps some of these chemicals that have been mentioned, like BPA or the phthalates, will have a different impact if we were considering their intestinal balance. Thank you.
I bet it's a comment, right? So
it's a comment, but perhaps somebody wants to volunteer some answer.
So we only have about 2.5 years of data collection on our project, yet we had discussions about exploring this and then ways to explore it. But probably not in the current project as it stands. But you're right, this is a evolving area of research that needs to be explored further. Thank you.
thank you. So we're going to take a question at the back of the microphone, and then please identify yourself, please.
Hi, I'm Athena Tippis. I'm at UCSF and Arizona State University. So my question is partially a comment, but I really am interested in what opportunities there might be in the ongoing research for addressing some of these questions. So there's an emerging body of research that pretty convincingly shows that psychosocial influences in early life have impacts on pubertal development, things like father absence, stress levels, etc. So why hasn't that piece been as central so far, and what opportunities are there to look at those kinds of questions in the data that's perhaps already being collected?
That data has been collected since year 1 in the study. So we've been collecting psychosocial data, including family composition, including the BASC, including depression scales in these girls for the last 7-8 years.
Okay.
And father absence. There's already been a paper published about father absence on this study.
Thank you.
Thanks.
A question from the front microphone, please.
Hi, Kim Harley from Berkeley.
Thank you.
I have a question for Dr. Bandera. I'm really interested in your measurement of the mycoestrogens, but I was wondering how much we know about the measurements that you're doing at this point. Do we know if you're taking, was it a single urinary measurement that you were doing?
Is it what?
How were you were measuring the mycoestrogens, I think, in urine? Do we know at this point how stable intake of these microorganisms is, or fungi are, and whether one measure is representative of long-term intake? Is this something that we know yet, or is this still emerging?
It's something we don't know yet, and that's something that we have to explore. Because it's so new. Again, this is the first study ever measuring and finding detectable levels. So that's something we want to do further studies to look at whether there's degradation, whether you have repeated measures, and how stable these compounds are.
Do we know anything about the half-life?
We don't know that much. We know in animal experiments, in pigs especially, we know that it has a half-life of 22 hours. But that's all we know. We know that also accumulates in adipose tissue. And it was interesting what I presented, that is an inverse association with BMI. So there's a lot of questions that we need to resolve.
May I have a question from the back of the microphone?
Hi, my name is Jasleen Goel. I'm a pediatrician. My question is, it came up from the legacy study, but it's generic. It's not really for a specific speaker. So it's two parts of the question. First is anybody studying boys that are born in families with family history of breast cancer, whereas the legacy study seems just to be looking at girls? And secondly, as far as I know, there's no genetic imprinting, whether the gene is inherited by a child from the paternal side of the family or maternal. Does anybody know different?
So I don't know about a specific study looking at boys with a family history of breast cancer. To my knowledge, anybody in the room?
Very rare.
Yeah, I'm not aware of any, and we are only including girls. Even so, there are boys in the families as well. And I'm not familiar with any other study.
Probably so rare that it will be difficult. Male breast cancer is already rare, so to have the family history and the male probably will be very difficult to study.
But wouldn't that be somebody who's at higher risk of falling through the cracks, a male child in such a family?
Yeah, it could certainly be something to be addressed in the future. I mean, these are important questions.
Thank you.
And one of the world's reigning experts on transgenerational epigenetic changes is our moderator. So perhaps our moderator should respond to that question.
Okay. I refrain. So may I have the question from the front microphone?
Linda Birnbaum. So I actually have kind of more just two comments. One is the issue about the mycoestrogens and the fact that they're really not acting like estrogens, they're more like anti-estrogens. And I think we've got to start moving away from these terms, because many, many different chemicals, their effects are tissue dependent. And I think we just have to understand that they're screwing up the endocrine system. So that may be a more helpful way to look at it. And the other thing, Frank, I think some of your stuff that you stressed on this distinctions kind of between the stages of puberty and how we're not seeing consistent changes in all the stages, like menarche, the drop in age for that is much less than, say, the drop in thelarche or stuff like that. You know, we're seeing the exact same things in boys, that again, you can break up pubertal development in boys and certain things are changing and appear to be impacted by environmental chemicals at different rates, that they're not all the same. And I just think it's something we might want to think about.
So thank you, because that allows me to say something that I couldn't during the presentation in response to your question. Because one of the things, so in looking at those life risk factors for breast cancer, those two studies by Algren and by Lee actually both investigated age of peak height velocity and age of menarche. They included both those in their models. And when you include both of those, age of peak height velocity actually supplants the variance that's supplied by age of menarche. In other words, it appears that perhaps age of peak height velocity is the more relevant variable. And from a biologic perspective, thinking about the age of menarche changing a woman's lifetime risk of breast cancer somewhere between 5 and 9% based upon 1 year delay in menarche just doesn't follow from an arithmetic approach to that issue, whereas age of peak height velocity is associated with a lot of pro-growth factors. And the earlier the age of peak height velocity in a girl, the greater the peak height velocity is. So it all follows why you can have an early maturing girl and a tall woman both have an increased risk of breast cancer based upon the same biologic mechanism. That seems to make a better totality to me. So thank you for that opportunity.
Thank you. I'm going to take, the next question is at the back.
John Yang from Great Neck Breast Cancer Coalition's Students and Scientists program. So we have increasing evidence of, girls maturing at earlier ages or just approaching puberty at earlier ages. And is there any possibility, or do you see any way that we can reverse this? Is this reversible? If yes, how?
Possibly. We have to better understand what are the underlying mechanisms. So if increased BMI is one of them, there's been a couple of studies that have looked at dietary intake. Those diets that have a higher consumption of fresh fruits and vegetables appear to delay onset of pubertal maturation a little bit. We know from natural history studies that those who have a higher level of physical activity have a somewhat delay in their pubertal maturation. And we know that both of those are factors which in the United States have gone the opposite way. We consume a more calorically dense diet with less fresh fruits and vegetables. Fewer and fewer kids endorse any regular physical activity. So those would be two things. Thanks, John.
All right, thank you.
Great job, John. Sandra.
Sandy Haslam, Michigan State University. So my question is relevant to the study of girls from families with or without breast cancer in their families. So one question I have is that are you considering, and I think you should, the different kinds of subtypes of breast cancers that exist within those families? And then also, given the age at which you're starting this study, it's not unlikely that some of the mothers, even in the non-breast cancer families, will develop breast cancer.
Yes, so we have information on the subtypes of breast cancer because we have collected extensive data from the moms that are in the breast cancer family registry. So we can look at that. And yes, we will update the family history during follow-up because you're right, some of the girls that are control girls right now, they might end up being positive for family history. So we will assess that.
Okay.
Thank you. I'd like to take the questions at the back.
Thank you. Now, if I'm not mistaken, I saw your charts of the different stages in a girl or a woman's life where she is most susceptible to developing breast cancer, and I believe they were but the different stages of life seem to cover every stage of life, and so I'm a little confused as to, is it just throughout her entire life she's very susceptible, or are there different points within these stages that, like, let's say a woman is more susceptible, like, when going through puberty than, like, let's say in, like, her mid-30s, for example?
Okay. And again, there's several experts in the audience who can probably respond to this question far better than I could, but I'll make a humble attempt at responding to this. So if you take a look at sort of If you remember the pictures that Dr. Fenton had shown, she was demonstrating some of the changes that occur within the breast tissue itself at these different times. And between the time of birth to the time of the onset of puberty is a relatively quiescent time. I mean, there's continued changes, but there's slow changes. And within the breast tissue, it's a relatively stable period. Then with puberty, there's a huge expansion in the number of cells. The cells are rapidly dividing, they're rapidly differentiating. The breast goes from what is almost non-palpable to something that is clearly visible. So there has to be a huge expansion in the cells, the cell types, differentiation. So that's very susceptible. That's going to be a period of susceptibility. It goes down a little bit, but doesn't resolve. And then with pregnancy, Dr. Russo will be discussing that this afternoon, I'm sure, is that there's another change that occurs in the breast. And so there's terminal differentiation of type 2 and type 3 into type 4 lobules. Am I?
Okay, good job. All right.
And so those are the times then, and again, okay, I'm sorry. So you may want to read the works of Dr. Fenton or Dr. Russo. They've actually discussed that and these periods of susceptibility. I am but a humble servant.
What I want to do is the last two questions is actually going to be between us getting hungry and the food. So, front.
Yeah, so Shauna Swan, I have a question about maybe a missed opportunity, or maybe you're doing this. It seems to me that insofar as there are familial factors that can affect the risk of hormonally related disease, this will maybe apply to the brothers as well as the girls. So have you thought about looking at the brothers? You've got the information in the family. You've got the information on the mother. Why not look at the age of puberty at the boys? Why not look at, you can go on and if you follow them to fertility, time to pregnancy, semen quality. I mean, there's a lot of things you can look at. But of course, I would look at, anogenital distance. But it's there's the other half, and I think we shouldn't be, it's important to look at sexually dimorphic disease patterns, definitely, but let's, when we study girls, think about the boys and vice versa.
Mm-hmm. Thank you.
I think that's a nice idea, and we could definitely do something like that because we're going to be in contact with the families every 6 months, and so we could build that into one of our follow-ups. So thank you, that's a very nice suggestion.
This will be the last question.
Bob Hyatt, UCSF. Dr. Biro, my friend, I think you said something right at the end of your presentation, which I want to highlight, or ask you to confirm, actually. So you said Menarche isn't changing that much, but Thelarche is, and so the time, at least between those two markers in puberty, is expanding, and you said, therefore, there's a longer period of time during the pubertal, for the pubertal period in which girls are exposed potentially to carcinogens and things that would initiate breast cancer. Now, that means that if you believe that theory, that you're less concerned about endocrine disrupting chemicals actually being the cause of the earlier age of Thelarche, perhaps, which is one way to look at it. So, what do you think we should be doing? Looking at the idea that endocrine disruptors are reducing the age of Thelarche, and that's the problem, or that puberty is increasing from whatever reason, the period of puberty is increasing for whatever reason, and we should be looking at the effects, carcinogenic effects of chemicals during this expanded period.
You want a short answer? Yes.
Okay, right.
Well, I think what we need to do is to give a big applause to all the speakers